this video is going to get you through the introduction to microbiology notes. I'm going to go pretty fast through this video, but remember you can pause the video at any point to make sure that your notes agree with what I have written on the screen. My tests do come off of my notes, and so you want to make sure that your notes are as accurate or as close to what I have up here as possible as we go through here. If at any point you have questions that I don't answer in this presentation, please feel free to email me, or you can Marco Polo with me, whichever way you want to play that game. Since this is chapter 1, I kind of wanted to give you some basic information. My name is Lori Heintz. My phone number, of course I'm not in my office because the campus is closed, but I still get emails if you leave a voice message, and so you can call me on my office phone if you want to. Email is probably the best way for you to contact me, or you can talk to me during the labs that we are actually going to be meeting on campus. You can also Marco Polo with me. I have posted some instructions for that in the updates within Schoology. All of the course is going to be posted in Schoology. You can get to that by going to the Hill College main page and then clicking on Schoology that's towards the bottom of the page. And then log in, and this is usually a person's login and then password so that you can get in there. You're going to have 2 courses when you go in there for micro. You're going to have a lecture section and a lab section. Make sure you're doing the work in both of those places because you can't pass the class if you don't do both the lecture and the lab. All the notes are posted in Schoology. I really recommend that you print them out and then read the book and fill in the notes, or watch the presentation and then fill in the notes. Again, all my tests do come off the notes. I was a person who took really good notes as I was going through school, and so I believe it's one of the best ways to study the material. If you have a better way, by all means do it, but that's kind of how my course is designed. This doesn't really apply since we're not meeting in person anymore. So the first thing you have is, what are microorganisms? Your definition is essentially up there. I want to talk about some of your examples. So bacteria is the plural form of bacterium. A bacterium is a single bacterial cell. So like E. coli is a bacterium, Staphylococcus aureus is a bacterium, but if you want to talk about both E. coli and staph, those are bacteria. Protozoans, those are things like amoebas and spirogyra and volvox and parameciae and a bunch of things that probably you guys haven't heard of unless you happen to have a dog that has gotten Giardia, or God forbid you've gotten that for yourself. Those are protozoan infections. They are made out of entirely different cells from bacteria, and they have to be treated differently because of that. Fungi are also eukaryotic, like the protozoans are, but fungi would be like Candida, which is the species that tends to cause yeast infections or thrush. Algae aren't pathogenic. They don't cause infections in humans, but they're very important for global oxygen cycles and carbon cycles. That would be like volvox and spirogyra. They're not quite plants. They're microscopic, but they are photosynthetic like plants are. And then helminths, or helminths if you would prefer to say it that way, that would be worms. So like tapeworms, roundworms, pinworms, those are also very often going to be microscopic. Although tapeworms can grow to be very large, a lot of the worms are actually very small. So when we talk about microbiology, most people automatically go to the bacteria. It's important that you understand there's a lot of other different things that count as microbes as well, and not all of them are pathogenic or cause disease in human beings. Who was Antony van Leeuwenhoek? He was actually a very interesting dude who I appreciate his scientific prowess. He improved microscopes at the time, which this probably doesn't look anything like a microscope that you've ever seen, but this is what he created. It had a better lens than anything at the time, and it allowed him to start looking at like droplets of water under his microscope. And what he noticed is there were things that would swim around in that water, and he called them animalcules. And so he's one of the first people who saw bacteria and made really good descriptions and drawings of what he saw under his microscope. My weird little side story that I'll give you about Leeuwenhoek is he was so in awe about the microscope that he developed that there was one story that he told where he was in the middle of having sex with his wife, and he stopped to collect his ejaculate and then view it under the microscope. So he's probably also one of the first people to see sperm under the microscope and then piss off his wife in the process. Robert Hooke is another one who was a more scientific person. He wasn't just looking at things because it amused him or he liked it. He was a scientist, and so he would peel bark off trees and then look at the bark. He would look at bread mold, which is what this is on this side off over here, and he would look at it. He was the first person to see a cell and call it a cell because he thought it looked like cells in a monastery, and so that name has stuck. We still are made of cells. Fungi are made of cells. But the microscopical mushrooms that he saw, that was actually the bread mold. This is Rhizopus, which is a species of bread mold, and so he was one of the first people who saw it and made drawings of it. The Origin of Microorganisms. When you look back at the fossil record of life on Earth, we have a pretty good record that shows that life appears to have first appeared about 3.5 billion years ago. And that life that first appeared was prokaryotic. Now what prokaryotic means is the cells are simple, they don't have a nucleus, they aren't as big as our cells, they don't have stuff like Ruffy, our Smoothie, our Golgi apparatus. Instead, they just have DNA kind of wrapped inside of a cytoplasmic membrane, and they still have cytoplasm, they just don't have all of those other parts. And so the earliest forms of life on Earth were very simple and very much like the bacteria that are still alive today. What is spontaneous generation is what you have next. So in the olden days, people believed that life appeared because God wanted it to, so God would create life. And again, you can believe that if that is how you want to believe, but hopefully you understand that humans don't just appear because God wants them. Two people had sex, they made a baby, and then the baby grew up. Well, prior to today's knowledge, people did just believe you could spontaneously generate life from non-life. This dude, he's one of my favorite dudes to try to read from back in the day. He made some very interesting ones. This particular one, he explains that if you get stabbed by a bayonet, in order for you to not die, you have to find the bayonet that stabbed you and then rub this salve on it, and that will magically heal you, essentially. But one of the other experiments that he did was he showed that you could supposedly create a live mouse by mixing hay and water, and for some reason his dirty underwear. I don't know why his dirty underwear was a magical thing, but if you mix those things together, you can make a live mouse. And so he believed that those were the ingredients for making a mouse. Again, hopefully you understand that that's not how mice are created today. They come from two adult mice having sex and making lots of little baby mice. And so we know now that life cannot be spontaneously generated. Maybe at the very creation of time, if you want to believe that, that's fine. But today, you can't have spontaneous generation of life. Louis Pasteur and Francesco Redi were some of the people who disproved the idea of spontaneous generation. So Francesco Redi was trying to show that some of the higher forms of life, like flies in this case, couldn't be spontaneously generated. And people thought at the time that if you just put meat on a counter, the meat would kind of turn into flies and maggots. What he showed is that if you covered the meat up, maggots wouldn't form on there, and so what was happening was flies were laying eggs on that meat, and then the eggs would hatch and give rise to the maggots. But if you cover it up and prevent the flies from landing, the maggots weren't spontaneously generated. And so he showed that you can't spontaneously generate maggots, you could only get them if flies laid eggs on that meat. Louis Pasteur took it a step farther. He went and showed you can't spontaneously generate microorganisms. So some things that he had already discovered were, if you filter air through a cotton plug, you can view that cotton plug and see organisms that had been in the air. And so there's organisms floating around in the air all around us. One of the experiments that we're going to do in lab is to allow you to leave an agar dish open to air, and you'll see bacteria and fungi growing on that after time, and that's just because those things are just floating around in the air. So people were saying bacteria were being spontaneously generated, and what Pasteur was showing is no, they're just floating around in the air, and so if you can prevent the air from contaminating a sample, the bacteria actually wouldn't get there. And to help prove that, he made this little thing, it's called a swan-necked flask. He would boil the broth within that flask, and boiling would kill any microorganisms that were inside of that broth at the time. By having this swan neck, bacteria aren't capable of walking up a hill and then coming in here and contaminating this broth. And so he could leave that broth that had been boiled on a table for years and it would stay sterile because the bacteria couldn't reach that broth. But if he tilted the broth to touch this little part where bacteria had settled, it would contaminate the broth. And so he proved that the bacteria were not spontaneously generated. They just contaminated the sample when you tilted it or allowed it to be open to air, and the air could settle bacteria into that broth. Contamination is actually something that we constantly have to battle in the lab, and that's, again, against that idea of spontaneous generation. If you end up with something growing on your petri dish, it's not because God hates you and he wanted something to grow there. It's because you contaminated that dish at some point in the creation of the agar, or you took the lid off when you shouldn't have, or something. And so it's important that you understand the world's not out to get you. Contamination is very important though, and it's also very easy to do. All right. This one is just for the majors. Pasteur did a whole slew of things. He did not just disprove the idea of spontaneous generation. So he is the guy who invented pasteurization, which essentially means that you take food products and beverages and you heat them up for a brief period of time and then rapidly cool them. That kills bacteria that are in them. It doesn't kill all of them, but it drastically reduces the number so that it allows that food to survive for a longer period of time. So it prevents spoilage. He also helped to develop a fair number of vaccines, that some of which are still in use today. And with Koch, they both came up with the germ theory of disease, which showed that, again, if you got a disease, it wasn't because God hated you. It was because you were contaminated with something and you developed a disease because of that contamination. All right, going back to everybody at this point. So John Tyndall tried to replicate Louis Pasteur's experiment with that swan-necked flask from earlier, and he was completely unable to replicate it. And so he was actually kind of providing evidence for that spontaneous generation thing, except that what they found out was because John Tyndall was working essentially out in the barn, there is something that we know today that they didn't know back then, which is that if you take hay or cut grass and you put it into water, you are always going to get a slew of growth that is in that broth afterwards. It's called a hay infusion. Now, because Tyndall was working essentially in a barn, his broth was being exposed to hay and all the things that were growing in that hay. And some of that stuff happens to be something that we now know today is called an endospore. And you can't boil an endospore and kill it. It's resistant to heat. And so even though he boiled his broth, he never actually sterilized it. Now, sterilized means you killed everything that was in that broth. Because he had endospores in it, he didn't kill it all. And so he got growth because he had not sterilized his broth to begin with. This again shows the importance of where you're working. And incidentally, it's why I don't think you can do a microbiology lab at home. Your home is not clean enough for you to do these experiments without contaminating all of your samples. You've got to have a lab setting for that. All right. Ferdinand Cohn went into looking into Tyndall's experiment, and he's the person who named those heat-resistant forms of life that Tyndall had contaminated his sample with. So today we call those endospores. This is Bacillus anthracis. It is the causative agent for anthrax, which is a respiratory illness, although there are some cutaneous forms as well. You can see that some of them are pink and they look smaller, and some of them are this kind of bluey color off over here. These are what are known as vegetative cells. They're active, they are currently multiplying, they're doing what bacteria do. These have started to realize, crap, we're running out of food, or oh no, it's starting to get too hot. And so they have done a form of binary fission followed by a couple of additional steps, and they have wrapped their DNA with 2 different cell walls instead of just one, and they've removed most of the cytoplasm. That makes them practically impossible to kill. It actually makes them very difficult to kill. You can kill them, it's just a lot harder to do that. And so these are endospores. They're not vegetative, these are vegetative cells. So Cohen is the person who discovered and named the endospores. All right. Normal flora. This is what's now known as the normal microbiota. I'm an old person and I was taught normal flora, so I'll refer back to this, but again, nowadays it's usually called the microbiota or normal microbiota. This is all the species of bacteria and protists and viruses and algae that live, not really algae, fungi that live inside of you that don't cause disease. They live in you and they actually help you out in ways that we are only just now learning about. They are very important for your health and well-being. So I'm giving you some of the things they do. They do way more than this though. So preventing disease by outcompeting pathogens. Well, if you already have 1 trillion bacterial cells living in your large intestine, and then you eat some egg that had fallen on the ground and you weren't paying attention to the 5-second rule or whatever, the bacteria that are on that egg are going to have trouble forming a colony in your intestines because you already had all of your apartments essentially full by the normal flora. And so they're outcompeting the pathogens for resources because they were there first. Those normal flora that live in both your small and large intestine, they also help you digest food that you can't digest for yourself, and in the process they make some vitamins for you, like vitamin K, that you don't get a lot of from the foods that you eat. Without this normal flora developing correctly, your immune system doesn't develop correctly. They help metabolize drugs for you. They help reduce the incidence of other infections. They do a whole bunch of other things that I can't even begin to discuss with you guys, but they're very important for your health and well-being. One of the inadvertent things that we have done with the advent of antibiotics and just giving them out essentially freely to everybody is we have really disrupted the normal flora. And it's been to our adverse health in terms of we now seem to be leaning more towards allergy and asthma and eczema, and it's because we have disrupted that normal flora. This I pulled from some different source. I don't remember where I got it from. I wonder if it'll tell me. Nope, didn't tell it. Did I put it up here? Nope, didn't say it here either. But this just kind of shows you some of the different species that you have living in different places in your body. You don't have the same species living in your large intestine that you have living in your nose for the most part. And if you get the species from your intestines into your nose, that's when you can have some problems. So everything lives in certain places or neighborhoods, and they're not supposed to move to other places. Your skin stuff stays on your skin and should not be in your intestines, and vice versa. All right. Other things that bacteria can do for us. They can help us to create a whole slew of foods that I happen to love. Like you would not have donuts and pastries were it not for yeast, which is a microorganism. It's a fungus, not a bacterium, but it is a microorganism. Beer and other fermented drinks, you get those again from yeast. Yogurt usually comes from bacteria. They ferment the milk, and in the process you get that whole live probiotic thing going on. Cheese and buttermilk, depending on the species, it can be a yeast or it can be a bacterium. It just depends on which kind of cheese you're making. Sauerkraut and pickles are usually bacterially fermented, and that's what gives them their interesting little snappy taste. Which of those should still have live microbes? The only one that should still have living microbes in them is the yogurt up over here. That's going to tell you it has live and active cultures. There should not still be live organisms in milk. In fact, milk is pasteurized because if it's not, it spoils very quickly. There are a lot of diseases that you can get from unpasteurized milk because it's not supposed to have live microbes, but it often does. The alcohol content in beer, wine, and especially liquors tends to kill off most of the microbes. So while you needed them to make it, when you consume it, there's no more live organism in it. Microbes that are being used in biotech. E. coli is used to make a whole slew of human medicines, including insulin. You can use other organisms like yeast, which is a eukaryotic cell, to make some of your more complicated things. And so E. coli can be pathogenic, but it's also part of your normal flora. This goes back to it's supposed to be in a certain place. If it's in your large intestine, you're usually fine, but if it gets in your urinary tract, now you have a urinary tract infection. That same organism that can sometimes be pathogenic, we use it to make a whole bunch of meds, and when they do that, they make clean meds very cheaply so that drug companies can make a slew of profit off of them. Pathogens are disease-causing organisms. These are the ones that people usually think of when they hear that term microbiology. But again, it's very important that you understand only a small number of bacterial species are actually pathogenic. The vast majority of them could care less if you exist or not. Infectious diseases or communicable diseases, those are diseases that can be spread from one person to another or one organism to another. This picture, I could have found a more modern one at this point, but this picture is from the 1918-1919 Spanish flu epidemic. I know you can't read this, but it says no spitting, so there are a lot of people in the world who believe that like if you cough something up, you should spit it out so that you can get the infection out. That's a wonderful way for disease to spread, and so they had to educate people that you should not be spitting your phlegm just out on the street because it spreads that infection. Once they learned that the flu was airborne, they even started to add like cubicle things or barriers between people. They hadn't figured that out at this stage, so this was essentially a wonderful breeding ground for the flu. Alright, next thing you have is what are some emerging infectious diseases. So emerging means they're new and they're just coming out. So Ebola's only been around since the 1970s, which I know you guys probably think is not that new, and yet it is. MERS is a coronavirus, kind of like the COVID that's going around right now, but it's Middle East Respiratory Syndrome. SARS is a severe acute respiratory syndrome, and COVID-19 is related to both MERS and SARS. Hep C, some people consider that to be an emerging infectious disease, some don't. Some of the newer forms of flu, like swine flu, are emerging. Lyme disease, again, it's only been around since about the 1970s. That one spread through ticks. AIDS has been around since the 30s, 40s, maybe even 20s, depending on molecular clock evidence, but it's really only been in the United States since the 80s. Mad cow disease is a prion disease. That one started in Britain because people were eating cows that were eating cows, which cows aren't supposed to eat cows. And so we kind of broke the food chain, and then people ate that contaminated meat, and then they got it that way. Zika virus is an emerging infectious disease that you guys might remember from 2016. That one spread through mosquitoes. There was a whole outbreak where there were a bunch of babies born with what's known as microcephaly, which is a small brain casing. Let's see, we've got chikungunya, we've got dengue. There's a whole slew of infectious diseases. COVID-19 is the newest one of those, but there's a bunch of other ones that you could put in there as well. What are some factors that help diseases emerge and spread? One, evolution. Flu viruses change every single year. It's why a person is supposed to get a different flu shot every single year. It's to deal with the new strain of flu that's going around. Sometimes pathogens can pick up new genes that allow it to become more disease-causing, and that's probably what's happened with the coronavirus that's causing COVID-19, is it picked up some new genes that made it deadlier than any of the other ones. The human population is ever growing, and so we've moved into areas that we didn't used to live in, and some of those areas had diseases that were present in populations of, like, bats. Bats are very good at spreading disease to human beings. Antibiotic resistance makes it harder to treat bacterial infections, and so that makes it easier for those to spread. Tuberculosis is one that is what's known as a re-emerging disease. We had it under control, but because it's becoming antibiotic resistant, it's now more difficult for us to treat, and then more people have it. This is from the Texas Department of Health, and it shows you the incidence of AIDS as of 2004 in the state of Texas. What are some of the factors that help this disease emerge or spread through the population? Well, there's evidence that areas that have poor sex education, in other words, places where they do like abstinence-only education, increases the rate of the spread of different sexually transmitted diseases, including AIDS. So by not educating students about proper forms of birth control that can protect against STDs, we actually increase the spread of those things. And so that's another factor. Education is huge when it comes to controlling the spread of disease. There are more people that have to take immunosuppressants now because of cancer therapy, or they have autoimmune diseases, and when they have weaker immune systems, it makes them more likely to develop infections. And then travel. You guys all hopefully know that COVID got from China to America way back in either December or January is what evidence is showing more, and that's because of people traveling to those places. This one is one that it should not be happening, but as vaccination programs work, people stop seeing those diseases out in the population. Like, when was the last time you saw a person with tetanus, for example? I had a friend of mine on Facebook just the other day saying she stepped on a rusty tin can lid and said, should she go get a tetanus vaccine, because she's never met a person with tetanus. And when you don't get those vaccines, it makes it more likely that that disease re-emerges. So this is mumps. We had mumps come through our area, I think it was like 2017, because of an unvaccinated population of Marshallese people in Keene. This baby has whooping cough. Whooping cough is another one that's re-emerging. And then measles, we had a re-emergence of that one in our area just a couple of years ago as well. Okay, so now we're going to switch gears and we're going to talk about the two major types of cells. A lot of people have a misconception that it's plants and animals based on what high school told them, but it actually is bigger than that. The two kinds of cells are prokaryotic and eukaryotic. What we're looking at here in this picture are examples of prokaryotic cells. Bacteria and archaeans are both made out of this type of cell. These cells are much smaller than eukaryotic cells. They don't have a nucleus. They do still have DNA, but they don't package it up like a nucleus. They don't have any of the other membrane-bound organelles, so no rough ER, smooth ER, Golgi apparatus. They don't have mitochondria. They don't have chloroplasts. They don't have vacuoles or things like that. There's just nothing with a membrane inside the cell. The cell itself does have a membrane, though. The cells that are in this picture right here, they are what's known as bacilli. That is rod-shaped bacteria. These happen to be gram-negative, but these are a decent example of prokaryotic cells. Next up, Eukaryotic cells is the next example. This whole big blue blob here is one eukaryotic cell. There's another one over here, and there's another one over there. So there's 3 eukaryotic cells in this picture. Eukaryotic cells are much bigger than prokaryotes. They do have a nucleus, usually somewhere towards the middle of the cell, although there's always exceptions to that middle part. There are other organelles too, like they do have rough ER, smooth ER, Golgi apparatus. They can have mitochondria and or chloroplast, depending on what kind of cell you're talking about. Just to show you the size comparison on this, so this is a cheek smear. Hopefully you did that in whatever freshman bio or A&P class that you took. So this is a typical eukaryotic cell that you could get from a human being. These little bitty specks on the surface that are dark blue, that's one bacterial cell, that's a bacterial cell, that's another one. And so you can see a prokaryotic cell is way smaller than a typical eukaryotic cell is going to be. Next up. This is just the book's picture that shows you a comparison of prokaryotic versus eukaryotic cells, and they really don't show that size difference that that previous picture did, but they're trying to show you what parts can be found in the cell. In order for it to be a cell, it does have to have a plasma membrane, which your book calls cytoplasmic membrane. It's the same thing. It's a phospholipid bilayer. Even prokaryotes use a phospholipid bilayer to make their plasma membrane or cytoplasmic membrane. Outside of that, there may or may not be a cell wall. It depends on what kind of subcell you have, like is it a plant cell or an animal cell. Plant cells do have a cell wall, animal cells don't. Almost all of the prokaryotes are going to have a cell wall, but there are, again, exceptions to that rule. In order for it to be a cell, it has to have some sort of genetic material. Prokaryotes just kind of throw it into the middle of the cell, so that region becomes what's known as the nucleoid, but there is not a membrane protecting that DNA. It's just kind of in a pile in the middle of the cell. Eukaryotes protect the DNA with a nuclear envelope to create the nucleus. There's very often a darker staining region inside of there called the nucleolus that you can't see in this picture. We've got rough ER, we've got smooth ER, we've got mitochondria, we've got a Golgi apparatus, so there's other organelles that are present inside of the eukaryotic cell that a prokaryote does not have. Next up, you have what are the 3 domains of life, and it's what's in the colored boxes. The book used to spell it wrong, but that should be with a K, so EUKARYA. The 2 prokaryotic domains of life are the domain bacteria, which includes bacteria, so like E. coli, salmonella, bacillus, anthracis, whatever. And then archaeans, that includes things that we're going to call extremophiles, organisms that live in places that are really hot or really salty, or places where you didn't think life could exist. Archaeans can live there. These 2, even though they are both made out of prokaryotic cells, they have very different rRNA, and they are not related to each other. In fact, there's evidence that the archaeans are actually more closely related to the eukaryotes based on their rRNA analysis. Eukaryotes are all in the domain Eukarya. This is where both plants and animals go. Fungi go here, and protists do. Whether or not this is a kingdom is up for debate at this point, but fungi is typically seen as a kingdom, and then so are plants and animals for that matter. So this comes up a little bit later in the notes here, where it says majors only. What is LUCA? LUCA is the last universal common ancestor. It is the ancestral cell or life that has given rise to all of the domains of life. Again, it existed over 3 billion years ago, over 3.5 billion years ago, and it gave rise to all 3 of those domains. So there are some features that they have in common. Again, like they all use phospholipid bilayers, they all use DNA as their genetic material. But they're, aside from that, very different from each other. And again, the archaeans and the eukaryotes are more closely related to each other than the archaeans are to the bacteria. And you can tell because this phylogenetic tree shows them branching more recently than the bacteria branch off here. So notice that this is talking about billion years ago. And again, the last universal common ancestor was older than 3.5 billion years old. This one would not have been in either of those groups because it was something that gave rise to all of those groups. Okay, let's see. You have, kind of looking to see where this was supposed to go, I guess kind of back into the 3 domains of life. You have what are they and what puts an organism into one of those domains. So we already talked about what they are. Next up, for the bacteria, that first domain and the one that we're going to spend the most time on here in this class, this group is entirely prokaryotic. You can find them anywhere. They're in the air around you, they're in your nose, they're on your eyelashes, they're just everywhere. Some of them are pathogenic, but by far most of them are not. Archaeans, these are the ones that I said tend to be extremophiles, so they live in places that are really hot. This, I believe, is Grand Prismatic Spring. It's one of the hot springs in Yellowstone National Park. The temperature of that water is crazy hot. Like if you were to just dip a hand in there, you would have regretted that really, really fast, because it would kill the cells on your skin that fast. It's that hot in there. I don't know the temperature though, right off the top of my head. We didn't think things could live in a temperature that is that hot, but it turns out after we cultured it and did DNA analysis, there are a lot of things living in there. And in fact, those living things are what give it the different colors that make up the wall. So things can live in really hot environments, really cold environments, really salty environments, even environments where there's no oxygen present. You can find some of these guys living. So even though they're prokaryotic, they're very different from bacteria. There is not a known pathogen that is in the Archaean domain. The domain Eukarya, everything in this group has eukaryotic cells. Some of them are single-celled, some of them are multi-celled. Plants, animals, fungi, and protists all belong to this domain. So, just questions that I usually ask when I'm doing a face-to-face class. Out of the two kinds of cells that we just mentioned, pro and eukaryotic, what do you think these are? Well, as you look at them, these were both taken with electron microscopes, which that's a pretty dead giveaway. It's probably going to be prokaryotic. But as you look at them, you can't see a nucleus, you can't see rough ER, you can't see any of those other organelles, and so these are both examples of prokaryotic cells. I just said this a second ago, but Archaeans are the ones that make the pigments in the hot springs, because it's a really hot place, and that's the only domain that can exist in those really, really hot places. Next up, you have what are the taxonomic groups and which group makes up a scientific name. This is usually taught pretty well in high school, but they may or may not have included domain depending on who taught it. So it's domain, kingdom, phylum, class, order, family, genus, species. For which groups make up a scientific name, it's genus and species. So running through humans for just a second, I was going to see if I had that in a slide. We're in the domain Eukarya, we're in the kingdom Animalia, we're in the phylum Chordata. We are in the class Mammalia, the order Primates, the family Hominidae, the genus Homo, the species Sapiens, and so our scientific name is Homo sapiens. As you go from the top to the bottom, things get to be more specific. So human beings are the only organism that are in Homo sapiens. But if you come back up here to domain, that's the most inclusive grouping. Like, we're in the same domain as a fish. We're in the same domain as an oak tree. We're in the same domain as an amoeba, because we're all in the domain Eukarya. And so as you go up, it gets more inclusive. As you go down, it gets more specific. How do you properly write a scientific name? This one's going to be very important because you're going to have to do an unknown report, and your unknown report is going to include the scientific name of the organism that you discovered in your unknown broth. Anytime you write a scientific name, the genus needs to be capitalized, the species should not be capitalized, and when you're typing it, it should be in italics just like it is right here. If you're writing it by hand, same capitalization rule, so capitalize the genus but not the species, and then you underline each word separately. For some reason, biologists, we are super OCD about don't underline the space. I've had points taken off a test back when I was an undergrad because I would just do one underline and then they would hit me for that. So I've turned mean just because my teachers were mean. Don't underline the space. Okay. What are fungi and which domain do they belong in? They are eukaryotes. They do have a nucleus inside of their cells. In terms of the microbial world, we're going to be focusing more on yeast and mold. Yeast can be pathogenic, so there are some yeast species that you can find in the human vagina, in the mouth. You can find it just in the perineal area, especially when you have a baby who has a diaper rash. That tends to have some yeast that's involved in it. Most of the species that do that, they are called Candida. That's the genus that you can find them in anyway. Molds is a lot more broad, so you've got like common black bread mold. You've got penicillin. Well, penicillin is the antibiotic produced by Penicillium, which is the mold that you can actually see here in this picture. So all of those are examples of fungi. Algae, what are they? They are essentially really, really small plant-like things, and most research at this point is showing that they really are plants and they should be in that kingdom. However, most books still put them into the protist grouping. This is Volvox. It's an algae. These are a couple of other forms of algae. So if you've ever had like little green blotches or filamentous threads growing in an aquarium, it's algae. Algae tends to be microbial versus plants are bigger and you don't need a microscope in order to see them. But these do photosynthesize. They use sunlight to make carbohydrates. They produce oxygen just like plants do. So microbial plants is kind of how you could view algae. Protozoans, on the other hand, are microscopic animals if you want to view them that way. They're not really animals, although some research says they are. I will kind of mention the taxonomy has been shifting a lot since the 1990s, and biologists can't agree on anything. So when I say things like they're sort of like small animals, it's because there are people who say they should be in the animal kingdom. I don't think your book does that yet at this point. I think they're still considering them as separate animals. So there's some ambiguity on where things get classified because, I don't know, biologists drink too much and we argue. We just can't figure out where we want to put it. This is trypanosoma. It causes a disease known as African sleeping sickness. This is an amoeba. It can cause something known as amoebic dysentery, and this is a paramecium off over here. All of them are protozoans. All of these are heterotrophic, which means they have to eat their food. They can't make it for themselves like a plant can. So again, these are microscopic animal-like things. What are helminths? Worms. This right here is a roundworm. It's known as a nematode. That is the phylum that you can find them in, is phylum Nematoda. Nematodes can be parasitic on especially plants. There are some that can infect humans as well, but they tend to be more plant pests than they do animal pests. That said, heartworms in dogs and cats, that's a helminth. That is a roundworm. It's a nematode just like this. Tapeworms are flatworms. They're in a different phylum. It's called phylum Platyhelminthes, but tapeworms are helminths as well. Even the annelids, like an earthworm, can be helminths, but we tend to not look at those because they're not parasites on humans. So the ones that we're going to focus on are going to tend to be the ones that are parasitic for humans or other animals. All right. Now, I did want to mention that I asked majors to go ahead and buy the non-majors textbook because it makes it very difficult to teach this class when there's two different textbooks being used. So essentially, you can just ignore the whole majors chapter 8 stuff, because I'm just moving it all into this one book, but I'm still telling them they have to know this stuff. So, next up you have, what are viruses made of? They are usually made out of two things: protein, which makes this little structure that's called a capsid. It's essentially a coat. And then they have some type of nucleic acid inside of it, either DNA or RNA, never both. Some will have an additional envelope that's made out of a phospholipid bilayer around that, but some won't. That just kind of depends on which specific virus you're looking at. For example, the coronavirus that's going around right now, it's an enveloped virus, and so it has that phospholipid bilayer that goes around it. Viruses can infect bacteria, they can infect plants, they can infect animals, so we're going to talk about viruses a lot. We're going to talk about them first in reference to how they can infect bacteria, and then we'll talk about what they can do to human beings as well. How do they survive is kind of, I guess, what I'll talk about on this one. So viruses are one of those organisms that straddles the line between living and non-living. They can't reproduce on their own. When they're not inside a cell, they show no metabolic activity whatsoever. So they don't respire, they're not doing a little bit of reactions. When they're not in something, they're not doing anything. But when they get into a host cell, that host cell starts to reproduce the virus. It starts carrying out the reactions that the virus wants it to do. And so they're essentially little cellular hijackers. How do they survive? They survive by making cells make copies of them. But once they're outside of the cell, they become completely dormant and hope that they find a new host. If they don't find a new host, they don't survive. But if they do, they continue that process of replication inside of a host. All right, did I tell you everything I wanted you to do there? Yeah, they live in hosts. We're good there. This is just the herpes virus. It is an enveloped virus. So this is the protein coat, the capsid. This is the envelope that goes around it. Herpes can cause cold sores. It can also cause genital ulcers, which is what I'm showing you here in these pictures. So it's a virus that is going to infect humans, and it has an envelope around it. Now, I use the term host there. A host, you can kind of think of it like the host of a party. When you're the host of the party, it's your responsibility to provide food and drinks and bathrooms and do what the guests of the party need to be able to do. Here it's sort of the same thing. If you become the host for a virus, you become a hospitable environment for that virus. You start making little virus babies, and you start providing it with the food it needs, and you remove its waste so that it doesn't accumulate. And so you become the host for that virus. Each virus tends to just have one or two species that they can actually host within, if you want to look at it that way. But sometimes they can jump between species, like there's evidence that the coronavirus jumped from a different species into human beings and then started to cause problems in us when it did jump. All right. Viroids are what you guys have next in the notes. Viruses don't have the protein coat that a virus has. and so they're simpler than a virus even. They also aren't either type of nucleic acid, they're just RNA, and at this point all we have found them in is plants, and so it does not look like we have any examples that cause disease in animals, just plants. This is potato spindle tuber. It's a little viroid that can cause blighting in a potato. This is the little strand of RNA that causes the problem. This is with an atomic force microscope, so it doesn't look like very much, but that's all there is to a viroid. It's just a little stretch of RNA. Prions, and that is how you say the word. I have met the person who invented the term. His name is Stanley Pressener, and if you invent the term, you get to decide how it's said, and so it's prion. Prions are infectious proteins. Prions are, in my opinion, the worst pathogen that's out there, and that's because for every single prion disease, there is no known treatment, there is no known cure, and they are all invariably fatal. And so basically, if you get a prion, it sucks to be you because there's nothing we can do for you. Prions also in a lab setting are very difficult to kill. The only way that you can kill them is to incinerate them, and that burns the entire thing up. You can't even kill them in an autoclave, which is how we traditionally kill bacteria in a lab setting. In terms of humans, the disease caused by a prion is called Creutzfeldt-Jakob disease, or just, it's French, so it's Creutzfeldt-Jakob, I think is how you're supposed to say it. Let's see, this one over here is scrapie. It is a disease in goats that you can find in our area, and if a rancher, or a goat herder, if you want to look at them that way, if they get a goat that has scrapie, they have to kill their entire herd. And traditionally, they would then dig a pit, pour gasoline on all the goats in the pit, light them on fire, and then bury them, and then not have goats for a while because you don't mess with prions. Now, coming back to Creutzfeldt-Jakob. Some of the first cases of this, actually, they are called mad cow, but in people. And it was because the way that we discovered it was people ate cows that had mad cow disease inadvertently. We didn't know the cows had the disease at the time. The prions transmitted from the sick cows into people, went through the gut-brain axis, and then essentially started to tear little holes into the brain of the people. So it creates a spongy-looking brain. That's why it's called a spongiform encephalopathy. It's because it makes your brain look like a sponge. Eventually it degrades enough of the brain that you just flat out die, but it sucks the whole time you're dying, and so just don't mess with prions if you can help it. I really already answered this question earlier, but there it is for the majors. You do still need to know your LUCA stuff. Let's see. Next up I have model organisms. These are usually something that we discuss in class, and I just wanted to make sure that everybody kind of had the same information. There's multiple places where you can look this stuff up, but I do still want to know that you have the right information that you looked up. So, what is the species name for the organism that causes C. diff colitis? It's Clostridium difficile. You can look that up so you can spell it correctly. Most people that get C. diff have recently been treated with what type of medicine? It's usually antibiotics. What happens is the antibiotics, when you give broad spectrum, it kills the normal flora just like it kills the pathogen. And unfortunately, C. diff tends to be antibiotic resistant. And so you've killed off a lot of your normal flora, but not C. diff. So C. diff starts to overgrow. It creates what's known as a pseudomembrane over the lining of your intestines, and then you get the worst diarrhea that is intractable. There is no cure for it in a lot of cases, and it is the worst, most foul-smelling diarrhea you will ever encounter in your life. And so it's one reason, yet another reason, why you should not give antibiotics to a person who does not need them. A new radical treatment is being offered to treat people with C. diff. What is it and why does it help? The newest treatment option is what is nicely called a fecal transplant. What that means is we essentially jam somebody else's poop up your intestines so that you get somebody else's normal flora in the hopes that it helps outcompete the C. diff to control it. And as gross as that sounds, there's two different ways that we can do it. The oral way, which means you're swallowing somebody else's poop, usually in a pill form. So you have to swallow usually about 30 poop-filled capsules that have been in a freezer, but as you're swallowing them, they're warming up and starting to smell like poop, so that's something to look forward to if you ever get C. diff. Or we can give you an enema, where we essentially just, instead of using water to flush out the system, we put somebody else's liquefied poo into your intestines. Either way appears to be okay. But it does help. Other antibiotics tend to make it worse or not even treat the infection, and so a fecal transplant can cure you where a whole bunch of other things really can't, so it is beneficial. Why is this treatment more effective than probiotic pills? So probiotic pills usually just have one or maybe two species of bacteria present within the pill, and there's no regulation of those pills whatsoever. In fact, there have been a few cases. So premature babies are given probiotics because their systems didn't have time to develop correctly. And there have been cases where premature babies were given contaminated probiotics where there was a fungus in there, and then the baby developed a fungal infection and ended up dying. So it's important that you don't take things that aren't regulated or you can't verify as safe through an outside lab. But even if it is safe, it still only contains one or two species of bacteria versus you have hundreds of species that are supposed to be living in your large intestine. And so it's a species count sort of a thing, and you don't get enough species with the probiotic pill versus if you get a fecal sample, you in theory get all like hundreds of species injected into you or swallowed into you, whichever way you want to say that. Next up, we have Plasmodium malariae. What does it cause? It's in the name, malaria. What is the vector? So a vector is something that transmits the infection but doesn't show any symptoms of that infection. And in the case of the malaria parasite, a mosquito, so something called an Anopheles mosquito is what spreads the malaria parasite. Is malaria living or non-living? It's living. It is what's known as a protist. We'll come back to that in a second. Which domain does it belong to? It is eukaryotic. Is it bacterial, a protist, or a helminth? It's a protist. Now, one of the reasons why I'm asking you that question is it's important to know exactly what is making a person sick, because you cannot treat a bacterial infection the same way that you treat a protist infection. Same with viruses. You can't treat viruses the same way you would treat bacteria. The same drugs don't do the same thing just across the whole entire wide spectrum. So you have to use antimalarials or anti-protist drugs to treat a protist infection versus you use antibacterials to treat a bacterial infection. This is a protist, and so you use an anti-protist drug. Will antibiotics clear up the infection? Okay, so it kind of depends on what you consider your definition of antibiotics. Classically, antibiotics means it's a drug that was produced by a living organism to kill another living organism. And so by that definition, no, the drugs that we use to treat this are synthetic. But if you think of antibiotics as just things that kill stuff, then yeah, you do use something to kill the Plasmodium parasite. It is not an antibacterial drug, though. Instead, we use what's been in the news a whole bunch lately, stuff like chloroquine, which is not a viral treatment, incidentally. It is a protist treatment, and it has really severe side effects, especially for a person that has heart problems. And so just kind of keep that in the background of your mind. Last step, HIV. What disease does it cause? AIDS, acquired immunodeficiency syndrome. How is it transmitted? Sex or blood? Is it living or non-living? Well, again, it kind of depends on who you ask. It is a virus. I believe that a virus is not living because it can't reproduce on its own and it doesn't do any reactions on its own. When it's outside of a living thing, it's inert. It's a particle. And so I think it's not living. We could argue about that if you wanted to in person, though, later. Is it a virus, viroid, or prion? It is a virus. Will antibiotics clear it up? No. Antibiotics will never clear up a viral infection. However, there are antivirals that can be used to treat some viral infections. In fact, there are numerous drugs that can be used to treat HIV infections. Where it used to be a death sentence, it is not anymore because we do have good treatment options. It's just that those treatment options are not antibacterial. They're antiviral drugs, specifically usually reverse transcriptase inhibitors. So that is chapter 1. I do want to warn you guys, so the quizzes that I have put to kind of prepare you guys for each chapter, they might ask you questions that we didn't talk about in the notes, but that's because those quizzes are open book, open note. You can look stuff up. The quizzes are a chance for you to get to know the material and learn about it in a little bit more depth. I do want to differentiate a quiz from a test. So the quizzes are what you have after each chapter to get you familiar with the material in that chapter and make you look at it in a little bit more detail. An exam, on the other hand, you do not get to use your books and notes on, but it does come more out of what I discuss in the PowerPoint and what I discuss in the notes, and so you won't have questions that weren't covered as we talk through things right here. So with that, go ahead and take that first quiz. You should be ready for it.